The overall goal of this procedure is to remove muscle tissue from a Drosophila larva in preparation for immunofluorescence analysis of larval dendritic arborization, or DA neurons, and epidermal tissue.
Today, we are going to demonstrate a method that improves visualization of neuronal and extraneuronal factors involved in dendrite morphogenesis.
The main advantage of this technique is that it permits immunofluorescence imaging of proteins that are otherwise obscured by muscle tissue, improves signal-to-noise ratio, and enables the use of super-resolution microscopy.
To begin, add enough cold saline to a silicone elastomer dish to cover the bottom surface. Add the subject larva to the dish and place it under the stereo microscope, ensuring proper placement of light. Position the larva with the ventral side up, which can be identified by the abdominal denticle belts. Stretch the larva in the anterior-posterior direction and, using insect pins, pin the head and tail to the bottom of the dish. With a pair of fine dissecting scissors, cut along the ventral midline of the larva from one end to the other. Spread the larva open and pin the cuticle tissue at the four corners so that it lies flat. Using forceps, remove the internal organs, including the CNS, gut, and trachea. Adjust the insect pins so that the fillet is taut but not maximally stretched. Locate the dorsal midline of the larva, where muscle tissue is absent, and then position a forceps prong at the interior of this boundary. Slide the prong between the muscle and epidermis.